Now, something happened to us in about mid-July, was that we were working on an orchard that, this is a cut deliciosa, I call it, because I'm not sure whether it's Bruno or whether it's Hayward. This is the rootstock, and this particular bit here is, I don't think, any relationship with PSA. It's what we call vine decay, and a lot of you will have seen this. But these little wee oozes here showed up where there was a grafted Hort 16A. And the next picture shows that bit of Hort 16A, this is only this big, that we've cut off, and when we cut it nice and cleanly, within 20 minutes you got this ooze, and it looked rather like toothpaste. And there's got a movie here to show you what happens in 30 minutes if you do 30-second. Okay? Do I click this or you? Can you click that? If nothing happens, we'll move on. Oh, it has. It looks like it's happening. I'm sorry. What's happening is that the ooze is coming up here. See this toothpaste ooze? It's all around here, and it's in here, right in the middle, just oozing up through the tissue. I think we've had enough of that one. Thank you. Makes me cringe, actually. And this is the little beggars that are causing the problem. This is they've got flagellum. They move. They're motile. And I hate this being called PSA Mike, but these are the little things running around, and it's just to show you that that's the enemy. That's the thing we're up against. Moves around a bit. No reason why it can't spread fairly well in tissue. Okay, so I've had enough of that one, thanks. This one gets more gruesome. This one is electron microscopy. And each one of these is a little PSA cell. My colleagues in Auckland do a very good job in this sort of thing. They've shown that these vessels in this particular Hort 16A we're looking at just chocker. There's just no doubt about it. Early on I had these pictures to show to colleagues and so on. We talked about them. We went, oh gosh, is that really happening? But it really is very strong. Okay, moving on. Where is PSA in the vines? Still a little bit hard to quantify after we've done quite a lot of work, but I just show you a few pictures about this, few slides. And we would take a vine such as this and mark off 10 centimetres with a little spray all the way up through, right away up here, right the way down. And we would take a sample with a drill, put it in a little wee vial, take it into the laboratory and test it. We would only test live bacteria. We're not interested in just dead bacteria, we're interested in the ones that really matter. And it's just got this one picture to show you of 5 orchards. This is the 5 vines in an orchard, and up here is the cane, and down here the trunk in each of them. And you can see very strongly up here where we often found an example or a symptom of PSA. Very strong reaction. This red is positive. The yellow is weakly positive, and the green, although I'm colourblind, is actually no PSA. And you can see that up here we've got no problem getting it from canes, particularly if they show a symptom, but the trunks have it as well, and they can be right down on the bottom of the trunk, and these are good positives down here. Now this is 3 out of the 5. It's quite tedious work, so we're working on actually 25 vines in various orchards to try and get some robustness into this. But as you can see, we've got PSA in trunks. The question of whether we have it in roots, and I know quite a few people have had a look at roots and they've found it in roots. I haven't seen any data about that, but looking at this, we started to look at some of our vines on the station, and this particular vine is a deliciosa rootstock, It goes right up to here. And this is a Chinensis scion. And we took the same sort of 10 centimetres all the way down here. And when we got down here, we had a lesion here. This is the reason why we did this. We couldn't find PSA in good-looking roots and with no symptoms. So we looked at one here where we had a lesion here. And the upshot of that is that when we diagram dramatic sort of presentation of this, there's that scion, that top bit up here, the 4 at the top. This is not detected here. When we got down to the trunk, not detected there until we got to the lesion. And then we have, this is weakly positive, but the important thing is that down into the roots, PSA was found quite low in the roots. So it's in roots as well at times. Okay, the summary is we looked at 25 vines in 4 orchards. 3 times we're doing this. We've done the winter one. We're now collecting that data. We're now doing the spring one. We're only interested in live bacteria. We found that live bacteria can be detected in wood using our technique. It's a tedious technique, but it's all the same. We could find it. Found it in canes, leaders and trunks. And also we found it in roots, particularly where there were cankers in the roots. Sometimes a vine is cut and a canker develops in the root before you know it, and perhaps that should be removed. Right. What happens in spring this year? We have been waiting for spring to happen to see further development of PSA, and it's happened. In one of our blocks, we've got so many blocks, about 50 blocks of kiwi fruit in the research station, and as I say, 54,000 vines. These are some of them. Just wanted to show you that some of them have survived here up till now, and a lot of them have been pulled out. As soon as they show any symptom in our research station, we get rid of them, but we've documented what material that was on. So we have copies of that material, so if we biff out a vine, we've got other copies of it. So this is tedious, ongoing, weekly. The whole of the station, everyone in the station has at least a block or 2 to do. And I think they're doing a fantastic job. We are finding blocks, we are finding this symptom, which a lot of you will be finding this year, the shoots collapsing. This is late September. They're actually being killed by the pathogen. It's in here somewhere, these ones here. Just another gruesome photo of it. It's a really good indication you've got PSA in your orchard. It's a really strong indication. Right the way down here. The rest of the vine can look perfectly normal, but you might find a centre of a cane with this in it. Here, just on the end. And so the summary from that work, spread in the orchard. PSA was active during summer, all summer. I've talked to you about the spots on leaves. They didn't seem to get bigger on the leaves, but we got more leaves with spots. PSA in wood, I've just gone through that. I hope just to let you know that I feel we've fairly well proved that this pathogen is in our trunks. In some orchards that are heavily infected, there'll be a lot of trunks. Less infected, less trunks. It's my view. Just a summary. Trepidation put this up. Because how to manage PSA in vines, I think it's fair to say no one solution fits all. We found it in leaves and canes and leaders, trunks and roots. It is less in Hayward. They may be slightly tolerant. So you've got to consider what you've got in your orchard. If you've got the rootstock that's Hayward, you know that's a decision you have to make whether you do these things I'm going to show you here. If infection is well established in the vine, you should remove the vine if you've got heavy infection. You might have to consider this as well. If the infection is light, you may choose to cut the trunk and regraft a less susceptible top on it. And this part is conditional on can they be identified. Some growers are choosing to rogue out any sign of secondary infection, obviously not leaves, but secondary infection, and they hope that they can keep a crop on, which I don't think is a bad idea either, if you're in that position to be able to do that. That means you've got light infection, very little in fact, is probably where people are faced with this, and they're carrying on. So I'd like to move over to the next person now. Thanks very much.